Welcome to All in the Field, AWS Agriculture Live.
On today's show, we have guests who are going to be talking about doing things that I didn't even know were possible, Karen. Today, we're going to be talking about the sunflower genome and Mauriceburg Lab and their contributions to the Registry of Open Data on AWS.
And that's the coolest part. There's so many new things happening in the world right now, and we should all be more aware of them.
Yeah, so let's go ahead and get started. All right, welcome everybody. I'm Matt Wolf, a solutions architect with AWS out of Chicago. I work with agriculture companies in the Midwest.
Excellent. And I'm Karen Hildebrand, and I am our head of solutions architecture for the agriculture industry. And I'm really excited today to be learning more about what's possible in the field of genomics. So let's head into our field notes today. Cool. So Matt, you know that I'm a nerd about fun facts. So I'm going to share my fun facts for today. And
Okay.
in a year other than 2020, about 4 million people go to the London National Gallery to see Van Gogh's sunflowers. But other random fact for you, my hometown is very near a giant likeness of Van Gogh's sunflowers, and it is 76 feet, 6 inches tall, and that's in Altoona, Manitoba. So there's your fun facts for today.
Wow, that's something. All right, I'll do you one better. Did you know that in 2019, over 225,000 metric tons of sunflower oil were consumed in the United States? Sunflowers aren't just a source of artistic inspiration. They're an amazing food source with tons of different varieties. In fact, I read about how AWS is helping researchers at the University of British Columbia understand how the different varieties differ at a genetic level and how we can use genetics to make the sunflower crop even more efficient and adaptable.
All right, you win. You out-nerded me on that one. Nice job. The amount of research in plant genomics is really fascinating, but it's also really complex, much more complex than the human genome or any animal genome. So the compute power needed to power that analysis is really quite significant. And the challenges are ever changing, especially when we think about the open source orchestration engines. And we have built ways for people to get started, including quick start guides, one that the moderators will be popping in here about Nextflow orchestration. But there's additional capabilities and open source orchestration engines that have blogs posted on AWS. We just won't be covering those in detail today.
Yeah, from a technical point of view, genomics really helps push the limits on what's possible and how fast we can kind of get things done, right? We saw with the vaccine development this year, which AWS has been involved in, and I think Ronnie or Nathan are going to be putting a link into the Twitch stream, into the Twitch stream right now for those that are interested.
Yeah, and certainly it's pretty amazing the acceleration of the capabilities. And we watched those all in real time this year. And it's a great way for those in genomics to really frame the problem and to really think about the number of concurrent processes that are needed and the capabilities and resources that can be spun up in a cloud environment enables significantly new approaches when it comes to plant genomics as well as breeding. And contributions to the AWS Registry of Open Data are really enabling collaborations between public and private sector in research in sunflowers, rice, and other commodity and crop types.
Yeah. Well, since neither of us are actually genomic specialists, we have a little bit of a treat here today. We have a couple of colleagues from AWS who are going to be joining us to co-host for that part of the show. Some of you may remember from episode 5, which was about geospatial data processing. Joe Flasher was here. He's with the AWS Open Geospatial Data lead team. Today, we've got the privilege of having one of his colleagues on today. So let's welcome Dr. Aaron Chu, who's also with the AWS Registry of Open Data. Aaron, welcome.
Hi. Thanks, Matt and Karen, for having me. And now I don't do quite as many puns as Joe, so you two will have to help me out. Hopefully I get my own sound effect. I don't know. But Thanks again for having me. I'm the life sciences lead here on the AWS Open Data team. And what we're all about on the Open Data team is democratizing access to data, right? And so that means helping folks learn how to access, share, and store their data smartly in the cloud. And we also maintain, as you mentioned, the AWS Registry of Open Data, or RODA. And this actually lists all the available data on AWS that researchers can access for free. I'm also a veterinarian with a specialty in canine genomics, and I have a PhD in epigenetics. So a lot of random dog information ready for you to make jokes about.
Okay, well, let's come up with something here. So I know, and I don't remember the answer to this. I really don't. But I know that like humans have 23 chromosomes. How many does a dog have, or a canine, I should say?
Well, Matt, maybe you remember that humans have 23 chromosomes because 23andMe is now a household name. But keep in mind that you can actually do the same thing with dogs and say 39 in canine.
Oh, so you
Perfect.
guys
I
are
know.
all
Yeah.
fun facts.
Today you learned.
Awesome.
And also, let me introduce my colleague, Dr. Jean-Sébastien Légaré, or JS. So JS is part of the AWS family now. But before then, he actually worked with our guest, Lauren Riesberg, and he led the technical pipelines for this first project that we're going to talk about, which is the largest genomic sequencing endeavor ever attempted in sunflowers.
For engineer
we
at
have JS
AWS,
on
and
mute. Oh,
We like
there you go.
reverse
Oh,
muted you.
oh, I'm
Sorry,
good.
JS.
I'm good. No problem. No
You're
problem.
You're
No
good.
problem. It's nice to be here and that you can hear me. I'm a senior software engineer at AWS, and I'm also a computer scientist at heart. And my background, my PhD, is in systems performance and cloud computing. And before I joined AWS, I had a wonderful opportunity to do research as a postdoctoral researcher in bioinformatics at UBC, University of British Columbia. And there I conducted multidisciplinary research with the Department of Botany with the Riesberg Lab. And it was a joint effort with the Computer Science Department and the Data Science Institute as well. So there, my focus was solving essentially technical issues associated with analyzing large genomic data sets, especially those of sunflower. And I want to welcome my supervisor at the time, Dr. Riesberg, to All in the Field. Dr. Lauren Riesberg does many things. He is namely the director of the Biodiversity Research Center at UBC, the BRC in Vancouver. And he's an expert in evolutionary biology. And at UBC, he leads a very large team of plant biologists that study the evolution of wild and cultivated sunflowers. So I'll leave the floor to Lauren.
Thanks, JS. It's a pleasure to be here, and I should mention that it was a delight to have JS in the lab. We really needed a true computer scientist. Most of the people who work in biology labs doing bioinformatics are trained in biology, and then they kind of acquire computer skills in sort of a eclectic way, often without any formal training, and certainly they don't know all the tricks of the trade that a computational scientist brings. So it was fun to have JS in the lab for a year. I only regret that he's not still with us.
I think that's our gain, I suppose. So thanks, JS, for joining us, as well as Dr. Riesberg. We really are excited to have, a variety of guests this year. We've had a lot of global guests, but full disclosure, I am Canadian, and I am very intrigued by the research that is happening within Canada. And so it's great to have this specific focus. I know my hometown was the site of Canada's agriculture research station for a large number of years. So it's great to be able to see what's happening in my home country.
Yeah, I was interested to hear that Karen was from Morden, Manitoba, because it has a storied history in sunflower breeding. There was one of the most important sunflower breeding programs was in Morden from the 1940s or 50s through the 1990s, and they introduced a lot of the resistance genes to important sunflower diseases, and the germplasm they provided also contained the components of what's now the hybrid production system for sunflower. And so basically all sunflowers that are grown commercially today have ancestry that traces back to material released from Morden.
Which is very cool. And I have to say, my sister and brother-in-law still grow edible sunflowers. Now my sister's going to think I disowned her, but it was really interesting in prepping for this show, just the diversity of sunflower production globally and understanding what the capabilities are there. So really looking forward to diving deeper.
Yeah, and let's get to it. Lauren, I always have to ask, how did you get started in the field of sunflower genomics? And I don't know if you guys are going to give me field as an agriculture pun, but I'm going to take it.
You should. It's a good pun.
Yeah, it's good. So I started studying sunflower genetics when I was and evolution when I was a graduate student, and I was making low-resolution genetic maps or genome maps of sunflower back in the late 1980s. So I was kind of doing genomics before genomics existed, so to speak. And that was at the point where the data sets were quite small, right? Just several hundred markers. And so you didn't require huge amounts of computational power. Of course, with the advent of high throughput sequencing in the 2000s changed everything. Instead of a few hundred markers, we can now interrogate all 3.6 billion base pairs of the sunflower genome. And so genomics became big data. And of course, that led ultimately to this collaboration with AWS. And you could see some of the wonderful variations in sunflowers there in that slide.
Yeah. Yeah, as we were just saying, sunflowers are so visually diverse. You think about like Van Gogh's sunflowers versus the sunflowers that we think of today, like what Karen's family has planted. Can you kind of help us maybe understand a little bit about some of the differences from then to now?
Yeah, so what's interesting is that one of my former postdocs actually cloned the gene and characterized the mutation that was underlying Van Gogh's mutation. It was his lab that did it, not while he was in my lab. And it's a mutation in a gene called cycloidea. And what this cycloidea does is it's responsible for making the sunflower petal. It's actually a ray flower. And the mutation that they found is actually just upstream or just beside the cycloidea gene. And what it does is it causes this gene to be expressed not just on the edge of the sunflower head, but throughout the sunflower. And so you get those ray flowers or petals growing everywhere. And that gives you the Van Gogh sunflower. It's sometimes also called in the sunflower community, it's called a doubled flower mutation. It's quite common. Also in the trade, it's sometimes referred to as the teddy bear sunflower because of the sort of fuzzy nature of the flowers. So in addition to Van Gogh's sunflower or teddy bear's sunflower, there are two kinds of commercial sunflowers that are primarily grown. One of them is the one that Karen was referring to in Manitoba in particular. There are typically confection sunflowers grown, and these are the ones with the large seeds and the white stripe down the seed that you see in a grocery store, and it's the ones that you eat. The other main type of sunflower are the oilseed sunflowers, and these have smaller black seeds. And both confectionery and oil sunflowers, when you see them in the field, they actually are fairly short. They're typically 3 to 5 feet tall. And this is quite different from the giant sunflowers that you see growing in your backyard. But these giant sunflowers are also sometimes grown commercially for biomass or biogas. And in Germany, for example, we often see these giant sunflowers grown near biogas plants. And the reason is that Germans prefer to see sunflowers rather than maize, which is typically used in the biogas facilities.
That makes a lot of sense. And that amount of complexity, and you think about that genome itself, I'm really curious, how do you use the underlying technologies like AWS has to enable the massive amount of genomic analysis in order to understand the different types of the sunflowers?
So our main problem before that pushed us toward AWS was that it simply took us too long to analyze our data. The data sets kept on getting bigger and bigger, and the one that we put on AWS is truly massive. And so it was taking us a year or so to run certain analyses, and it was just too long. It was prolonging projects for graduate students or postdocs. And so it just wasn't acceptable. So then we turned to the cloud, which allowed us to complete our analyses in a more reasonable amount of time. We were able to massively parallelize the analyses, and JS will talk about that a little bit. And also, another good thing about the open repository on AWS was that it gave us a place to store our data in the same place that it could also be used. And that's a huge time component, is just transferring the data to one storage component to another.
Yeah, that's right. The one fundamental shift that the cloud allows is that you don't really have to scrounge for resources the way you would normally have to do it in a university setting, where you have to book a few servers on the compute cluster. In the cloud, you can do one machine running for 1,000 hours, but that's roughly the same cost as 1,000 machines for an hour, or 2,000 for half an hour. So that's sort of the big difference there. And another thing that's a bit more subtle about the cloud is that it lets you put labels and tags on about just any piece of data that you generate. So anything you compute for an hour, you can give it a URL, you can go visit it, you can share it with people very easily. And we, as part of the work in the Riesberg Lab, we built a framework on AWS that lets you, it's called Bunnies. It allows you to reproducibly compute large-scale genomics experiments. So it's a pipeline framework.
Okay, I've got to ask, why is your pipeline called Bunnies?
Yeah, I know. We use it for sunflowers, but it's called Bunnies because they reproduce a lot. So it was all axed on reproducibility. So there's Lauren kind of briefly touched base on this, which is that in academia there's a lot of rotation between students and projects, and over the years you end up reusing some of the results from previous former students or former researchers, or other teams even in other universities, and it's very challenging to know exactly all the steps that were involved in producing a large data set. You spend thousands and thousands and thousands of dollars into sequencing, aligning, calling your data set, and you have to know how it was generated, what software was used. And so that framework essentially is a way to capture provenance. Every piece of data that is generated, you can go from there and see what all the raw inputs were used to generate it. And so if you spot a problem later on in your pipeline, you can kind of go back and see all the files that were affected by this change.
Very cool.
Yeah, for us, it's Bunnies has been really useful, not just in capturing that sort of information about exactly how it was done, but one of the things that we find is that we're constantly producing new and better reference genomes and new pangenomes that we use, and then we want to take all of our data and reanalyze it again. And it just happens repeatedly. And so having a pipeline like Bunnies makes all of this much more reproducible and much easier going forward.
Well, that's great insight about how you guys kind of set the systems up to do that analysis. But after you've kind of now started looking at the data and ingesting it and doing computation on it, what kind of insights from the sequencing projects kind of have stuck with you the most?
I think the one big thing is scale. Essentially, for a lot of the analyses that we had to do in the Riesberg lab, the data sets grow over time, and the more data you include in your analysis, the more sort of statistical confidence you gain in your results, the more resolution you have. And it comes at a point where when the compute time you need to spend to analyze these data grows exponential with the amount of data you have, you realize, sooner or later that the fixed set of resources that you have in your university or your lab or won't cut it, or you'll have to exponentially wait longer to get the results. And so I think the cloud is really where it shines, where you can, in practice, kind of sort of assume you have as much compute power as you possibly could want. The other place where, again, scale plays a role is that if you are to fit, it really is the only place where you can imagine having, every single plant genome fit inside one system. So it's something that's referred to as the pangenome, having a reference that encompasses all the possible variation in a species or across species. And I think that's where the cloud really can come in as a hero in the story.
Oh, that's good to know. So let's talk a little bit more, Dr. Riesberg. Sunflowers can really kind of grow in some harsh conditions. Can you kind of walk us through some of what we've got a visual here that we'll talk through.
Yeah, certainly. So part of our interest in sunflowers is that they're often found in extreme habitats, and they're also cross-compatible with domesticated sunflowers. So you can basically mine genes from these wild extremophile species to breed more environmentally resilient or more climate adapted sunflowers. And so one of the big discoveries from this AWS Open project that we did with JS was that we found that the genes, we discovered many of the genes that were actually underlying these adaptations to these extreme environments, so they can be directly targeted by breeders. But what was both interesting and challenging is that many of these were actually inherited as non-recombining blocks of genes, or as what we call in the literature supergenes. And in most cases, these supergenes were formed by large inversions or large other structural rearrangements or structural variants. And so it's interesting because these supergenes could be employed by sunflower breeders to sort of more rapidly adapt sunflowers to extreme conditions or to various stressful conditions. But they also represent an important challenge to sunflower breeding, not just to us in terms of us analyzing them, but also to sunflower breeders, because these blocks of genes not only contain the genes that the breeders want that might make, for example, a sunflower more drought tolerant, more tolerant to low nutrient conditions, but they also contain genes that the breeders don't want, but they're inherited as a block. So it's going to be very difficult for them to be sort of those blocks to be teased apart and to pull out the individual genes. So it's both a interesting finding, but also a challenge.
So the next question then is for Aaron. The Registry of Open Data on AWS contains the genome from the University of British Columbia in terms of what they've contributed in terms of the sunflower data set. It's updated twice a year. It's available in an S3 bucket. Could you tell us why the Registry of Open Data on AWS exists for these types of genomics projects and what that looks like for others to use it?
I sure can. And as I said earlier, we on the Open Data team really believe in democratizing data access in the cloud. And I think that for the life sciences and genomics in particular, where you have groups working on similar projects and really accessing the same sources of public data, this program and registry is just so important, right? So we developed the Registry of Open Data, or as we affectionately call it, RODA, to be a catalog of this publicly accessible data on AWS, all stored in Amazon S3. So these data sets are maintained by data providers who come from all over the AWS ecosystem. And that includes academia, government, nonprofit organizations, startups, and biotech, pharma, larger corporations. And along with information on where and how you can access the data, as Karen said, that these all live in public S3 buckets, and so you can actually access these right out of S3. RODA is going to allow the community to curate and provide tutorials, usage examples, publications relevant to the data. And that's the data provider and the data user. So basically, we're helping data users and data providers form an ecosystem. We call it a community of practice, where the efforts of one person, regardless of if they're making the data or they're using the data, is going to benefit the whole community.
Okay, so then Dr. Riesberg, what's some of your hope for how this contributed data set is being used? Are there like global collaborations, public, private use? You know, what's also the benefit to the Riesberg Lab for contributing to some of this data?
Yeah, so our data is used, we expect it to be used globally and certainly has been in the past. Many of the public and private breeding programs have obtained germplasm from us that contains alleles from or genes from wild species, but also a lot of our data is being used to, we actually formed a public-private sunflower consortium to do genomic-based research. In this particular data set on AWS, we think it will be most useful for creating sort of environmentally resilient sunflowers or climate adapted sunflowers, because we've actually identified genomic regions or individual genes that we know, for example, lead to greater heat tolerance or allow or greater drought tolerance and so forth. So we imagine that those will be targeted first, but also any large data set like this contains contains enormous amounts of variation, and companies are very interested, for example, in disease resistance. So I'm sure they'll also target the data set to search for potential disease resistance targets as well. And in terms of our benefits, you know, we work closely with a lot of both public and private breeders, and we have many projects with them, and so we benefit from their success. And we also the AWS provides a very nice way to store our data for free, because that's one of the real challenges we have is actually keeping these large amounts of data and storing them and keeping them in a way that they're available for us to use.
Great. All right, I think we're going to move on here to the next segment, viewer comments.
All right, this is always fun. Oh, here's my attempt at terrible handle reading. PH Fan 1987. How big would the pangenome be?
Oh, that's an interesting question. So in general, the pangenome, you know, we actually don't know for sure at this point. We know for genes, the pangenome is probably going to be about twice the size of sort of a single genome. But if you add in all the repeated elements and other elements of the genome that are not shared between types, I wouldn't be surprised if it's 10 times the size of an individual genome. So 3.6 gigabases, and I mean 36 gigabases instead of 3.6 gigabases. We don't know for sure yet, though.
Cool. Second question we got here comes from Nathan 007. Lauren said that the hardiness. What's that? Oh yeah, the easier one. Right. So Lauren said that the hardiness genes could be crossbred for traits in other sunflowers, but what about breeding with other crops for drought tolerance?
So at this point, one could use genetic engineering or CRISPR-Cas approaches to target particular mutations we've discovered that are associated with drought tolerance, for example. But right now, you obviously can't cross a wild sunflower with corn or something. But I think the information can be used. And you know how that information will be used is again sort of something that will probably be controlled as more by socio-political concerns than by technology.
That makes sense. Well, thanks for those couple questions. Let's dive right back into the rest of the content, because I know there's lots to share here. So, Erin, I think you'll take us back in.
I will. Let's dive back in. And I wanted to bring up another project that I know, Lauren, your team has been working on, one that's aimed directly at aiding local food and crop banks.
Yeah, so let's see. Yeah, so oh, I guess I was supposed to wait.
No, that's no problem. Yeah, so can you just tell us a little bit about how you're involved in DivSeek International?
Yeah, so this was something that came out of a meeting that one of my graduate students and I organized about a decade ago. We were interested we got together a group of people who were working on the genomics of crop wild relatives with the interest of sort of trying to tap the diversity of wild species in crop breeding. And one of the sort of main conclusion that came out of this meeting, a main challenge we realized was that we needed an open database that to catalog all of the genomic and phenotypic diversity found in the world's seed banks. This is where all germplasm from crops and their wild relatives are being maintained. And initially at that meeting, we were super ambitious. We were thinking that we should actually sequence them all ourselves, form a consortium to do it. And then we became a little less sort of more realistic, I suppose. And we did publish a paper in Nature sort of outlining what we thought should be done, which was basically to make a database. And this effort was led by Susan McCooch at Cornell University. She was one of the participants that helped organize that original meeting. And the DivSeek International now, it's now a legal entity in Canada led by Susan, and it focuses on developing and guiding standards that people should use in gathering and sharing information and in the development of databases.
Awesome. And I do have to call out Cornell. I am a 3-time Cornell alumna. My first degree was from the College of Agriculture and Life Sciences. So, heartbeat a little faster, happiness there. But now with my role on the open data team, one thing that you did mention, Lauren, was gold standards for how people should be developing databases with the objective of sharing data. And what I see personally with a lot of my data users is this struggle to access and then harmonize data across different databases coming from different sources. What strategy did you take to harmonize the data sets that are compiled in DivSeek?
Well, yeah, I mean, this is the perennial challenge, right? This is why we thought there should be a single database that everything was on it, but it turns out people aren't. They want their data in their own thing. And so what we focused on really was to make make sure that the different databases can talk to each other. And so that's the key thing. And also we asked that our members ascribe to the FAIR principles. This is that makes information findable, accessible, interoperable, which is the most difficult thing, and reusable. And we also sort of provide, I guess, soft moral pressure for those, and encouragement for those who are not making their data easily usable and accessible by others. There are a lot of different personalities in the field, and some people are very comfortable with sharing everything, and others really aren't. And that's what we see in daily life as well. So it's no different in genomics.
I definitely agree. And this isn't really agriculture or even genomics specific, but do you have any words of advice for researchers out there who are trying to aggregate and reuse data?
I do have a few things to say about that on the technology side of things, which is that especially in terms of aggregation, putting things in the same bucket, in the same basket, technology is making things easier for sharing and making things public and giving accessible URLs to things. Other than that, when you have your data in your data store, your data lake, the other thing you need to do is tagging the data properly. If it's not tagged, annotated, no one is going to use it with very high confidence. And so this can go down to like defining an ontology for your data, like what exactly do you mean by seed size? How do you measure your seeds? The seed color, making sure everybody's using the same, palettes, and defining if you align something or if you do variant calling, defining what references you've been using to perform these computations. Um, and so figuring out when you have access to data, whether you can use it, does also require that the methodology that you used in your experiment, both what happens in the wet lab and what happens in the computer, you need to include the program versions that you used, what parameters you used, and you need to have potentially like a search, make it data searchable, or have an index to figure out, access things quickly. Yeah, that's what I'd say on the technology side.
Yeah, so I would agree with JS that metadata submission is super important. In seed banks, they call this the passport data, and this provides information about any individual seeds and where they came from and what the environment was like and so forth. And often they have phenotypic information about those seeds and so forth, and that's really, really critical. A goal that we have at DivSeq and so forth is to actually link genomic data to the physical germplasm. Everything with a DOI, a digital object identifier, and so that any type of genomic data can go back to the actual seed accessions that a plant breeder can use and can order from the seed banks.
That is really interesting, especially thinking about the storage constraints and then having that passport data lineage. It's really interesting to think about how that can be used in genomics. But Matt, let's bring it back to the field.
All right. Our last few minutes, why don't we touch on what many consider to be the genomic revolution, and which warranted, I think, the Nobel Prize in Chemistry this year as well, genome editing. So Dr. Riesberg, how is genome editing and specifically CRISPR-Cas9 leaving its mark on both sunflowers and more broadly, I guess, on the agriculture industry?
Yeah, it's not been quite as revolutionary as we had hoped. In sunflower, at least, there was a lot of interest in gene editing at first, but then this was really dampened by a recent decree by the European Council of Justice, that Court of Justice, that is equating gene editing with GMO, with genetically modified organisms. And sunflower is a non-GM crop. This is a conscious decision made by the sunflower industry to target the vegetable oil market in Europe. And so as a consequence of this decree, I do not think we'll be seeing gene-edited sunflowers in the field anytime soon. And this is a real shame, because gene editing, as you can see in this figure, offers a means of crop improvement that's far faster and more precise than that can be achieved via conventional breeding. So with CRISPR-Cas9, you can actually target individual genes and mutations, whereas with conventional breeding, you actually are moving chromosome fragments which contain many genes. So there's no way it's going to be as fast or as precise. Thus, if socio-political concerns, I should call them, sort of generally do not prevent the use of the technology, then I think the impact on agriculture will be huge, and it will play this discovery will play a huge role in sort of ensuring food security over the next century. I do worry about how broadly the technology will be used because of sort of these other issues that are entirely non-technical.
And I think that kind of leads into something we talk a lot about at AWS, which is seeing around corners, seeing what's coming, and having a thought process that helps us to remove some of those blockers. So, as a leading researcher in the sunflower genome, what do you see around the corner, 3, 5, 10 years out?
Well, one of the things that's clearly sort of, is that every project, every plant will be relying on a pan-genome rather than a single reference genome. But also, I think that the data sets are simply going to be getting larger and more challenging to analyze. And as a result, I think a lot of researchers will be moving to the cloud, even more so. So right now, I think you have 45 data sets or something. In 3 to 5 years, it's going to be 4,500 instead, or something like that. I think it's going to grow exponentially. I do think, in terms of the other prediction would relate to gene editing. I really do think that political factors or socio-political factors, regulatory factors will largely stymie the commercialization of gene-edited crops, and I think it's a real shame. And so I think genomic data will primarily continue to drive conventional breeding and making it faster and more precise rather than seeing this gene editing revolution.
There's also a trend that's been going on is that it's about building blocks, especially in your research analyses pipelines, is that the common operations that many researchers, many industries need to do end up being commoditized. So they get turned into building blocks that you can use for your own pipelines or turning into services. So that's something that I think is going to keep happening.
Yeah, I think that's a really good thing, because right now the field, and this is with databases too, Aaron, I mean, the field is just replete with reinventing the wheel. We're all doing the same thing in different groups in a slightly different way, and it makes no sense. Instead of reinventing the wheel, people can use what's already available, and I think that's one of the things that's going to happen over the next decade.
Great. All right, we're going to do one last check-in here with our viewers while we still have Lauren, Aaron, and JS here. So let's see if we've got anything in from the chat stream. Well, we have one question, I see. I think this one's for Aaron. How complex is the dog genome compared to Lauren's sunflowers?
Oh, well, we can't pit flower against dog here. It's worse than comparing apples to oranges, is what I think. But I think JS mentioned this either today or yesterday. There's an assumption that because plants maybe aren't snuggly and responsive the way dogs are, that animal genomes are more complex than plant genomes. And it's actually quite the opposite. Yes, dogs have more chromosomes, but the kind of structural diversity that you can evaluate in the plant genome, any plant genome, is going to be more than what you see in your average animal or mammalian genome, actually. Usually with animals, if you see something like a loss of function mutation, right, something that breaks a gene, you're going to see the animal have to compensate. That's going to probably take a few generations. Or unfortunately, right, you're going to see one of the many inherited diseases that we see pop up in domestic dogs, for example. Plants can sometimes just say, oh, look at that, and then they'll just make a new set of chromosomes. It's actually quite incredible what plants can adapt to much more quickly than animals.
All right. Well, I want to say thank you to Lauren. We're going to be moving on here to our next segment, field tips.
Okay, thanks all very much.
Thanks.
Thanks,
All right,
Lauren.
since we have JS here with us today, who has done some phenomenal work on genomics project prior to joining AWS, we asked him to share one of his tips with us here today.
Do I have 1 hour for this? 5 hours or 5 minutes? I could talk a long time about this. I have a lot of experience migrating existing bioinformatics pipelines that were written in a very ad hoc way into the cloud, and some quick tips I can talk about are that you have a very large data set. You're very likely that your experiment, you're going to run it once at the end. You don't want to pay 3 times for running the same experiment. So it's very important that at the end, when you look at your data later on, you're able to kind of trace back your steps. So don't skimp on annotating your data, figuring out what data set you've used, your software, your parameters. You will also have very likely, potentially months of trial and error to make sure your compute's working the way you want it to, so that you're getting the results you want. And so it's very important to use version control, using Docker containers to, making sure you get the same compute environment every time you run it. Detect cases of corruption. It's bound to happen. Mislabelings in your samples, being able to go back and figure out, what needs to be recomputed. And if one thing that you'll have to do is try to anticipate the errors that you'll get later on as you throw in more and more data into your set. And what is bound to happen, you'll get things like, timeouts, corruption again, or just machine errors, hardware errors, things like this, storage failing. you'll have to be able to rerun certain parts in increments. And so you want to be able to reuse partial results and annotate these partial results correctly. And, things you'll have to annotate is like the software versions you use, the short read archive versions you use for your raw sequences, species information, command line parameters. You might also want to know how much it costs to generate a piece of data, so how long it took to compute. This will help you realize whether you should, keep a copy of it in store or recompute it as on demand later on. Your runtime environment, all the extra third-party software that you've pulled in as part of dependencies. So what that translates to for your existing programs is breaking down your existing scripts into multiple smaller compute units. Each of these units need to be able to restart them independently. What that will let you do is also run these chunks in parallel. So you'll actually end up speeding up the whole process in that, doing so. And you'll eventually realize too that when you split up your programs into multiple chunks and you run them in parallel, you'll end up with pieces of data that are reused in all of these chunks, like your reference genome. So you'll also have to think about where to put these data items that are constantly being retrieved over and over and over and over to save money. And you'll have to think about, if you think of your pipeline as going in phases, if you're always doing the last phase, then maybe you don't need to keep all of the data for the previous phases around. So you'll need to think about cleaning things up as you go. Those are very few of the things I could recommend, but I don't want to take all the time.
This is my favorite part about working at AWS, is that there are experts like you and Aaron that we can tap into that know so much more about any given topic. And I'm so glad you work in S3, because clearly there's a ton of data storage in your tips and how to make sure that you're tagging your data appropriately. So thanks for sharing your tips. Really appreciate your joining us today.
Thank you. It was fun. Have a good day.
Thanks. Bye.
Great. Well, thanks to JS and all of the other guests that we had on here today. Another big shout out to Nathan and Ronnie for answering questions on Twitch for us live. They've been sharing hopefully some resources in the chat that everybody can make sure that you're keeping a look at. Looking forward also here to next episode next week, All in the Field, where we're going to be live streaming across the reInvent channel again on Wednesday with Scott Fagenbush of Deere's Intelligent Solutions Group.
Absolutely. It'll be a really great show. We'll be talking about their approach with collaboration with other agricultural companies through the Develop with Deere program, including a startup, FarmDog, founder Lyron Birch, who will be with us as well.
Yeah. So thanks for all the interest here in agriculture. We're going to be doing actually 2 shows next week, evidently. On December 16th, we're going to be on at 3:30 p.m. Pacific time, and again at 6:30 p.m. Pacific time.
Absolutely, because we don't want to leave our agriculture animal interest group out. We want to make sure that we have something for them, as well as our controlled environment agriculture spectrum as well. So we are going to be focused on animal feed production to improve animal rumination. So fascinating all around.
Yes. So please do join us live next week for our last 2 episodes of the season of AWS All in the Field Agriculture Live.